Imagine you go out and sample two populations. You want to see how different they are, so you sequence their DNA and compute FST. Now, there are many ways to do this, but consider this approach. You can translate normal multi-locus sequences into single locus data by considering each sequence with unique identity to be a distinct haplotype allele. Then, you can compute FST on that multi-allelic locus. How are your choices of how to construct those alleles going to affect your computation of FST? You could imagine that the longer the sequence you translate into an allele, the more alleles you get. This is important because depending on the diversity of a locus, the values that FST can take are constrained. In fact, this is well described. Only when the frequency of the most frequent allele is 0.5 is FST able to take on its maximal value, and FST is bounded if the frequency of the most frequent allele is higher or lower. We can start with a very short sequence and progressively add more SNPs and ask how FST changes. Adding a SNP that doesn't provide totally redundant information does two things. 1. It increases diversity and therefore can move the frequency of the most frequent allele to the left. And 2, it will affect where FST occurs within this new bounded space. If you add a SNP with a low degree of differentiation between the two populations, it always decreases FST, unless it doesn't actually change the haplotypes. On the other hand, if the SNP we're adding is itself highly differentiated between the populations, we might expect FST to go up. After all, we're adding information about the populations being different. But actually, it can sometimes also go down, in part because the SNP affects not just how differentiated the populations appear, but also the maximum possible FST value they can assume. So, this becomes sort of hard to generalize with multiple SNP additions, so we consider a simplified case of adding additional loci at a certain frequency in leakage equilibrium. What happens? You'll need to read the full paper to find out. So, what does this mean if you're actually using haplotype SNPs to compute FST? Well, the biggest takeaway is that you have to be careful in constructing these haplotypes because the choices you make can hugely impact the results. For example, if you're trying to figure out which pair of populations is least differentiated using real data sets among a group, you may get one ordering with one size haplotype and a different ordering with another. If you want to learn more, check out our recent paper, now out in Genetics.